Good afternoon, everyone. I'm Colin O'Connor, and I'll be your moderator today. The title of today's webinar is Unraveling the Roles of Copy Number Variants in Tumorigenesis, and it's sponsored by Thermo Fisher Scientific. Our speaker is Xiaolan Fang, Associate Director of the Cytogenomics and Molecular Pathology Laboratories at the Henry Ford Health System. And with that, I'll turn it over to Dr. Fang.
Thank you, Colin. Good afternoon, everybody. I'm thrilled to have this opportunity to present some of the clinical research data from Henry Ford system and share that with you through the invitation by Thermo Fisher. So thank you. The title is Unveiling the Roles of Copy Number Variants in Tumorigenesis. And I have no financial disclosures or conflicts of interest in relation to this presentation. So this is just like a brief list of the lecture objectives. I hope we can explore the evolution and development of cytogenetics and cytogenomics through history. And also, hopefully, the audience will gain some familiarity with the current tools and techniques used in cytogenetic analysis. Also, we want to understand the unique advantages and benefits of OncoScan in comparison to other array or NGS platforms for cytogenomic analysis in the research. And finally, I hope after the presentation, you would gain some knowledge about the specific types of tumor samples that can be effectively analyzed using OncoScan technology. So first of all, let's just go through a quick, brief review of the clinical diagnostic process. Usually it starts with the sample collection and then the sample preparation. And also the final step is sample analysis. And for the sample analysis using genetic testing, we usually cover those by the work done by three major labs: molecular lab, cytogenetic lab, and the biochem lab. So for today, we are more focused on the cytogenetic lab work, that is for the microarray, and also we'll do karyotyping and FISH in addition to microarray and cytolabs. And for molecular lab, most of the tests perform the sequencing assays and the non-sequencing assays like MLPA or Fragile X, which is related to the triplet expansion tests. And for biochem lab, the major analysis focusing on the metabolic analysis and enzyme analysis. So a little bit history of the development of cytogenomics. In 1888, the term chromosome was first coined. And in 1923, human reported to have 48 chromosomes. And in 1947, the term karyotype first appears in PubMed. And interestingly, in 1956, the human confirmed to have 46 chromosomes. In 1970s, the chromosome bending technique developed, which opened a new door for all the cytogenomic knowledge about chromosomes. And in 1986, FISH, or the fluorescent in situ hybridization, was the beginning of combination of molecular technologies. When it started to be used for cytogenetics, it is using specific sequences of DNA labeled with a fluorochrome to determine if that sequence was present or absent on the chromosome. In 2002, the first bacteria artificial chromosome array, CGH, started to be used in clinical labs. And the first clinical array actually has 287 probes covering the entire genome, while in 2004, the oligo array, CGH, and SNP array copy number analysis started to be used. In 2007, CMA, or the chromosome microarray analysis, is reported to be cost-effective compared to karyotype and FISH. And then in 2010, ACMG and ICA recommend replacing karyotype with chromosomal microarrays, and followed with the recommendation array design, analysis, and performance in 2012. And in 2014, after the multiple publications in 2013, in 2014, CytoScan DX was cleared by the FDA for usage in diagnosis of patients with development delay, intellectual disability, and congenital anomalies. And in 2020, ACMG and ClinGen published the recommendations for the technical standards for interpretation of CNVs. So through the development of cytogenetic techniques, here is a summary of all the units of resolution, and you can see it follow a specific pattern for increasing this resolution. So for haploid genome, the approximate size is around 3 billion BP, and the whole chromosome is around 50 to 250 million BP, and those could be detected by standard karyotyping. And when the bending technique was developed, we can search for like specific chromosome band at 400 to 550 band stage, or higher with the high resolution bending to up to like 850 band stage. So the 400 to 550 band stage, we can detect around 5 to 15 million BP, while the high resolution one, we can go 1 to 3 million BP. And the next step is the submicroscopic region that could be detected by FISH or like comparative genome hybridization or chromosome microarrays. So those could narrow down to 50 to 250,000 BP. And when the whole genome sequencing was carried out in the recent decades, nucleotides detection cut down to 1 to 1,000 BP. So you can see through all those techniques, the development of cytogenomics from chromosome to chromosome bands, genome loci, and whole genome analysis. We always start from the big picture, just with low resolution at the chromosome level, and then genomic loci level, increase the resolution, and finally we have the whole genome analysis. So we have high resolution of the whole picture. And interestingly, if you compare it to the development of molecular genetics and genomics, which I go through briefly here, it's a little bit different. So in 1590, microscope was invented. It's unclear who invented the first microscope, but it was generally agreed that two Dutch spectacle makers and father and son team, Hans and Zacharias Janssen, created the first microscope. And in 1665, Robert Hooke published a book called Biography, a book filled with drawings and descriptions of the organisms he revealed under the recently invented microscope. And in 1839, cell theory was developed, which states that all organisms are composed of one or more cells, that cells are fundamental unit of structure and function in all living organisms, and that all cells come from pre-existing cells. A major milestone in molecular biology was the discovery of the structure of DNA. And this work began in 1869 by Friedrich Misker, a Swiss biochemist who first proposed a structure called nuclein, which now we know to be deoxyribonucleic acid, or DNA. And it's generally recognized that the chromosomes were first discovered by Walter Fleming in 1882. And then in 1930s, the molecular biology was established, followed by the genetic codes discovery and the central dogma in 1950s. And in 1990 to 2003, the Human Genome Project was established and completed. So if we compare to the cytogenomics development, the molecular genomics starts from like a small portion of the big map with high resolution, like a single gene, to a panel of genes, and then to the whole exome or whole genome sequencing, covering the whole the whole genome, the big picture with high resolution. So despite the differences between cytogenomic and the molecular genomic techniques, they lead us to the era of big data. That is, we have a large amount of the data. And with such amount of data, how can we place together and the information and rebuild the biological network at molecular level and to understand the mechanism and to facilitate clinical diagnosis, risk prediction, research, etc., is our challenge right now. So back to microarray, which is the focus for the talk today. Let's talk a little bit further about this technique. Chromosome microarray analysis, or CMA, can be divided into two major categories. First is a two-color experiment, also referred to as array comparative genomic hybridization, or ACGH, in which patient and normal samples are differentially labeled and hybridized to probes on a single microarray. And second is a one-color experiment, also known as single nucleotide polymorphism, SNP-based microarray, in which instead of including a control sample as a reference in every run, reference intensity data has been built from a population of normal samples and used as a reference for the patient sample. The two-color analysis generates results as a signal ratio, while most of the one-color assays provide the absolute signal intensity information. The ACGH consists of CNV probes, while the SNP arrays have mainly SNP probes. So we have more details in this figure. In this slide, you can see the two types of CMA listed here. Panel A, or the figure on the left, represents ACGH, and the figure B on the left represents the SNP array. So in a two-color experiment, DNA from individuals of the same species or different tissues from a single individual, for example, normal and diseased cells, is extracted and differentially labeled with compatible fluorophores, for example, CY3 and CY5. Equal amounts of labeled DNA is hybridized to the microarray, and the hybridization reaction is allowed to come to equilibrium over a period of over 12 hours. At most probes, equal amounts of the two samples will hybridize yellow features on the array, reflecting the fact that most loci in the two genomes are present in equal amounts. Regions that are deleted in the sample genome, for example, region 1 of sample A, will result in probes with increased relative C3 signal at the screen features here. Alternatively, amplified regions in the sample, region 2 of sample A, like here, will result in features with an increased relative CY5 signal, or red features. Over the entire microarray, the signal ratios at each feature follow a Gaussian distribution, and the candidate copy number variations are identified on the basis of the deviations of a particular probe ratio using statistical cutoffs. Although the diagram illustrated the protocol for an array comparative genome hybridization, all array procedures, including SNP discovery and insertion site mapping, are carried out in this way. And one-color experiments, which is the panel B and the figure, are performed in a similar manner, except that the DNA is labeled with a single color and hybridized to a microarray without a reference sample. The difference between two-color and one-color experiments is that the former case, two samples are compared within an experiment, while in the later case, two separate experiments are required to compare the samples. So here is a summary between those two methodologies, and you can see the probes, a little bit different. One single sequence oligonucleotide of around 60 BP for ACGH, while two 20 to 60 BP oligonucleotides of different sequences for SNP arrays. And then like two labeled DNA for ACGH, and one only patient DNA labeled and hybridized for SNP arrays. And the resolution down to size of the oligonucleotide, exon by exon, but no detection of UPD or consanguinity, or under the limit SNP addition possibly like in the ACGH, while for SNP arrays, the resolution limited by SNP distribution. It is able to detect the consanguinity and most UPDs, and also the detection of most known clinically relevant CNVs, but not exon by exon. All right. So for OncoScan microarray, which we'll talk about today, it has incorporated the technique called molecular inversion probe, MIT technology, to generate targets, which hybridize two probes on the OncoScan platform. So the genomic DNA is hybridized for 16 to 18 hours with CNV and somatic mutation probe mixes, which is represented here. And it's recommended, like usually you need 80 nanogram genomic DNA by the manufacturer. But in published literature, these labs suggested like as low as 30 nanograms could be used from the FFP extracted genomic DNA. So this varies among different groups, but basically 80 nanogram is the starting point, as we recommended. And for the molecular inversion probe here in this chart, you can see different steps through the procedure. For step A, that's the annealing, which probe and the genomic DNA hybridization. B is the gap filling with AT or GC nucleotides. C is an exonuclease selection for gap-filled probes. D is cleavage at site 1 for probe opening and inversion. E is the probe amplification and biotinylation. And F is the cleavage at site 2 to release the tag sequence. G is the array hybridization followed by staining with phycoestrine through the biotin and streptavidin interaction. H is the final step, which is array scanning. So here you can have a quick look about how it is done step by step. All right, so for MIP microarray, several research groups suggested that it is great for FFPE and fresh frozen samples. And the reason they recommended it is because the copy number performance is really good. As I highlighted here, you can see the ratio for past QC and the ratio for the high quality data collection from the ones that passed the QC are really high. Most of them are over 50%. A lot of them are nearly like 100%. So this is really dramatic improvement compared to the other techniques using FFPE. And also they have a good performance for the genotype detection. As you can see here, the medium genotype inconsistence or the range genotype inconsistence are really low. So they basically rule out all the inconsistence for the genotyping. So those are two major features that they use for recommendation due to the MIP microarray technology. So as a summary, we can see the SNP arrays is really powerful, especially the high density SNPs. And then they can do a lot of things, including confirming the copy number events, breakup point determination, UPD copy neutral loss of heterozygosity detection, triploidy detection, genomic contamination and identification, low-level mosaicism detection, sample heterogeneity, clonal diversity detection, allele-specific changes or hyperploidity detection, Mendelian consistency and checking, and parent of origin studies, testing. So these are all the possible applications for the high density SNP arrays. All right, so we talked about some background information. Now let's move forward and talk about some actual samples. We'll start with somatic samples because OncoScan has a great advantage of treating solid tumor from the FFPE treated procedure. So we mainly will focus on that to start. All right, we'll start with brain tumor. And this is a sample with glioblastoma or GBM. And one particular feature for the glioblastoma based on CNVs is the 7 gain and 10 loss. And based on WHO guideline, 7 gain and 10 loss is enough to support a diagnosis of the GBM based on the CNV platform. And also you can see other features such other CNV markers like EGFR amplification or homozygous deletion of CDKN2A. We also see other amplifications and deletions based on the array data, but those will be the major features that we use for the GBM type of samples. So this is another patient with GBM, and you can still see the 7 gain, 10 loss. And in this patient, the CDKN2A deletion is heterozygous instead of homozygous. And at the same time, we can also see the loss of 13, 14, and the 22. So those are the additional CNV marker, CNVs detected in the sample. All right. In addition to the 7 gain, 10 loss, during our experience handling the GBM samples, we also see some variants which are not exactly the same as the 7 gain 10 loss, but similar. So we call them like variants. For example, in here, we can see 3 different samples from the individuals with GBM. They all carry the 7 gain, but for 10 loss, it's a little bit different. They could have normal copy number mutual, but like loss of heterozygosity on chromosome 10, or they could have like partial loss of 10, like here, and similarly here. So they are not completely carrying the loss of 10. They definitely have the gain of 7, and those are not exactly the same as the classic marker for 7 gain 10 loss, but we still have put them in a part of the GBM. It's more like a variant type. And similarly, for some of the patients with GBM, the samples will show a 10 loss, either complete or like partial. But chromosome 7 doesn't always necessarily have a gain. So on the top, you can see this one has a normal copy number of 7, and then partial gain of 7, and a small portion of gain of 7. So those are all kind of variants, and we're still figuring out whether those variants are related to the level of the aggressiveness of GBM in the individuals. All right. So the second type of CNV tumor that we want to talk about is the astrocytoma. This is an example of grade 2 astrocytoma. Morphologically, it's grade 3. And as you can see, this patient has homozygous deletion of CDKN2A and several other CNVs. And when the astrocytoma develops to grade 4, usually they will have a molecular marker that is IDH mutant. But they also have other things like loss of chromosome 13 and 14. And in this sample, we also see chromosynthesis of chromosome 19. And also, interestingly, for this sample, we didn't see the loss of TP53, but we do see a copy-neutral loss of heterozygosity of 10.5 megabase existing on 17p, including TP53. And while we do the concurrent NGS sequencing for the sample, we identified a pathogenic variant in TP53, R273C, which explains that this one is a homozygous mutation based on the CN level H status. So that would add to complete the story of the sample and an analysis. All right. So one type of specific astrocytoma is pilocytic astrocytoma. And this type of cancer actually have a very unique marker. That is the 2.0 megabase gain at chromosome 7q34, which is here. And this will cause to the breakpoints located in KIAA1549 and BRAF, and consistently with a known tandem duplication event. So this is the common fusion arrangement involved in the this 7q34 duplication, where you can see the long arm of chromosome 7 is here, and then the on 7q34 the tandem duplication actually results in a fusion of the BRAF and the KIAA1549. So the fusion between the 5' end of KIAA1549 and the 3' end of the BRAF gene that codes for the kinase domain. So the fusion gene will code for the BRAF kinase domain together with the N-terminal part of KIAA1549, replacing the BRAF regulatory domain. So it's pretty unique, and then it would activate the downstream signals regulated by BRAF. And here they show the FISH results for those. This is the normal cell. In the tumor cell, you can see the fusion signals. All right. So the third type we're talking about in the brain tumor is the oligodendroglioma. It also has a very unique and typical marker based on the CNV features. This is 1p19q co-deletion, as you can see in this figure. And for this sample, we don't see a lot of the other CNVs, while in some other samples, in addition to 1p19q co-deletion, we also see the amplification of this spot, and then also like this is the 7q, the gain of 7q, and loss of 14q. So those are like the additional CNVs. And the fourth type of brain tumor we want to talk about today is meningioma. So for meningioma, the histopathological progression from grade 1 to grade 3 are listed here. Grade 1 is typical, grade 2 is atypical, grade 3 is anaplastic. Those are subtypes would become like representative, and when the tumor become increasingly aggressive, the cellularity would increase, and together with the nuclear atypia formation and loss of cytoarchitecture. So for grade 1 meningioma, usually it's not very happy with CNVs, as shown in this sample. It's basically normal. And this is a grade 2 meningioma sample. As you can see, they have a representative or pretty recurrent CNV that is loss of 22. And this is another one. It doesn't have a loss of 22, but it does have like a lot of the copy number increase and decrease. A lot of them are gains, and it's almost like nearly a triploidy. When we move to the grade 3 meningioma, we can see the CNVs increase a lot. So in addition to loss of 22, we also have the CDKN2A and B loss, and also other like gains and losses. Basically, it's just make it more complicated, and I think that represents or associated with the increase of the pathogenic level. So this is probably the other type of the tumor. After the brain tumor, we see a lot of skin tumor samples here at Henry Ford as well, and then they have pretty distinct CNVs for melanoma. So this is a sample for the CNVs for nevus, and it basically is normal. You didn't see much of the like CNVs in this pad. However, when we check a melanoma sample, you can see a lot more of the CNVs. So basically, increased number of CNVs instead of particular type of CNVs is more common in melanoma samples. And usually larger than 5 CNVs will be observed in melanoma samples. So the next step, next type of the solid tumor we're talking about is the renal cell tumors, which we also get FFPE samples and will check their copy number variant related to status. So here is a nice publication in 2019, which they listed the molecular and the cyto features for each type of renal cell carcinoma or RCC. They listed those for clear cell RCC, like papillary RCC and the chromophobe RCC. And we'll talk about the clear cell RCC and a feature they have, that's the chromosome 3p deletion. So this is a sample with the benign neoplasm in kidney, and you can see basically it's normal, nothing really standing out. But when we check the clear cell RCC sample, we can see a lot more CNVs, including the 3p deletion, which is listed in the literature. That's here. So that's most of the that covers most of the somatic samples that I want to talk today. And OncoScan could also be used the constitutional samples, but this is not focused today, so I'll just go through this pretty quickly with some research publications. So in 2021, Wen et al. Published their data about OncoScan microarray assay and saying like they could improve the abnormality detection rate in POC samples. So they basically could have like 2% increase for the normal karyotype, 26% increase with the culture failure, and 57% increase in FFPE samples from the POC. And this is the flow chart they displayed in their publication for the procedure. So POC start with the cell culture, and if the cell culture is good, it will go through the karyotyping, and then the karyotyping will have like 50% abnormal expected fully, and then the other 50 will go to the POC normal karyotype, and then together with the ones that have the culture failure and FFPE, they'll go to DNA extraction and go through the OncoScan microarray assay. And they could, most of them have some positive results. If there's no definitive results, they will go to the POC non-karyotype or like no pathogenic finding section, and they will go to the exome sequencing. So for cytogenomic arrays in constitutional and somatic testing, usually they perform with higher positive diagnosis rate or like the positive results comparing to G-banded chromosome analysis and FISH. And here is just like a pretty brief summary. So 3% detected by G-banded chromosome analysis, 3% by subtelomere FISH analysis, 7 to 11% detected by targeted arrays, and 18 to 20% detected by whole genome arrays. Those are focused more on the constitutional samples for the children with, or infant and children with developmental delay, intellectual disability, and or multiple congenital anomalies. All right. So overall, I think I summarized the samples and the advantages for microarray in both somatic and a constitutional setting for research. And here is a little bit comparison for the advantages and the limitations of cytogenomic arrays. So for the advantages, they are far more superior to alternative methods for detecting copy number variants that are below the resolution of G-banded and chromosome analysis. Basically, it's like a high resolution technique. And then they could detect tandem duplications as sensitive as for the deletions. And also, unlike karyotype analysis, CMAs do not require dividing cells. So that's a huge advantage if we're treating with the FFPE samples. Because FFPE samples originally developed for pathogenic use, they meant to like be stained and then with either like IHC or like H&E. For the DNA extraction, they are not the optimal samples that we would prefer to use. So for limitation of cytogenomic arrays, they cannot detect the balanced rearrangement, such as like inversions, balanced insertions, and Robertsonian translocations. Those are more like the unstructured variants, and they can detect more CNVs, which are VUSs, or like variant of unknown significance, which would probably increase the complexity and like analysis time for each test. And they cannot detect the low-level mosaicism, which usually are below 10 to 20%. And then they cannot detect the single gene point mutations. All right. So, but for the OncoScan, there are also like pros and cons compared to other research arrays. Mainly here is the other like platforms provided by Thermo Fisher. So it's like CytoScan HD 7500K array. They will have like ultra high density analysis of chromosomal aberrations, CNV and LH. And then the outcome a segment report with identified aberrations. And for CytoScan Optima, or mainly focused on the prenatal samples, they will have the detection of aneuploidies, submicroscopic aberrations, and mosaic events. And they are able to identify maternal contamination, triploidies, and copy-neutral events such as absence of heterozygosity and uniparental disomies, like UPD and CNLH. And the outcome is the segment report will identify the aberrations as well as for those three. And for OncoScan, its strength is for the samples with poor quality, especially for like the FFPE, that they can do the determination of copy number changes and allelic imbalances, including loss of heterozygosity in solid tumor from small amounts of highly modified and degraded FFPE-derived DNA samples. So I believe that's the end of my presentation, and I want to send my acknowledgements to Dr. Brandon Shaw, who is a Senior Director at Henry Ford Health System. Brandon is a great mentor and the leader, and he provides a lot of encouragement and guidance during my time here. And he is truly a great colleague to work with as well. And I want to thank Dr. Abir Tabara, who is the neuropathologist here and provided most of the pathological support for all the CNV, the CNS tumors, the brain tumors, and like other solid tumors for the pathological analysis and grading information. And I want to thank Jessica Sanchez, who is the senior lead technologist in our Cyto lab, and she provided a lot of technical support for the microarray analysis and procedures. And also, of course, the awesome team from Henry for the cytogenomics lab and the molecular pathology lab. And lastly, I'm very fortunate to work with the Department of Pathology and the Laboratory of Medicine, where we have a great team of the pathologists at Henry Ford. And they gave me really good learning abilities and knowledge-based support for my work related to the research, the clinical research projects here. So thank you all for all your support. And that's the end of my presentation, and I'm ready to take questions. Thank you.
Great. Thank you very much, Dr. Fang. We can now start the Q&A. So the first one we have here, besides the CNVs for pilocytic astrocytoma, is there any CNV pattern in CNS tumors that could be explained by a specific molecular/cytogenomic mechanism?
Yeah, that's a good question, because a lot of people will ask whether the CNV patterns are associated with specific molecular mechanism. So pilocytic astrocytoma is one good example. And another example that I mentioned here with my presentation is the oligodendroglioma. The 1p19q co-deletion actually is suggested to be related to the whole arm translocation. So I think that could be an explanation. And also for some of the solid tumors, actually it's like based on the CNV number instead of the particular or specific CNVs like we see in melanoma. So it really depends on the tumor type and on tissue type.
Great. Second question here. Does an increase in CNV number and complexity and the complexity of CNVs always correlate with a higher tumor grade?
The short answer is yes. I think with the examples I show, the CNA ranged from normal to really highly complicated level. Those are pretty typical in majority of the solid tumors. But like if you are already at the higher level, like for GBM, as the grade 4 is the highest for the glioma, I think it's just adding more CNVs probably will represent a more aggressive pattern, but it probably won't change the prognosis much because the prognosis is already pretty poor. So it really depends on the stage of specific tumor and then the type. Yeah.
I have one more question here. So if anybody in the audience is on the fence about sending one, send it in now. For the GBM samples, are the variants of +7, -10 associated with similar aggressiveness of the typical +7, -10 or different?
That is a great question, and that's actually the key point for our research project here. Because when we are searching for the variants, not like the molecular or cytogenomic variants, but the pathogenic variant types of the GBMs, we're trying to focus and correlate and see whether those are specifically associated with the aggressiveness and like the progression stage for each sample. So I think that's a good question. Yeah, hopefully that answers the question. Yeah.
Is OncoScan a good tool to assess HER2 amplification?
I would assume so, but like it's more costly. And currently we have very good FISH assay platform to detect the HER2 and as well as IHC. And I think those are more compatible with the FFPE slides. Those are very established methods. So I would say yes, but it's really luxurious to use OncoScan for HER2 amplification detection. Yeah.
Got it.
Thank you.
How can you classify deletion in an oncogene or duplication in a tumor suppressor gene? Can they be likely benign?
That is a good question. Personally, I would think this is a case-by-case scenario, because if it's an oncogene and it gets deleted in the tumor, usually we don't think it much. Like if it's a pure oncogene, it doesn't have any like tumor suppressing functions. However, in real world, the role or the molecular regulation based on each gene is really like complicated. If you say like you deleted an EGFR in the tumor sample, whether it's helpful for tumor, probably it won't be considered as like a oncogenic event if it's a deletion. If it's amplification, yes, for sure. So I think it's just something like it really depends on the specific gene's function and the specific type of cancer. And then like we know for some of the genes, they are regulated more frequently in one type of cancer, but not the others. And in that condition, I think we would put it as a VUS for like likely benign if it's definitely just a tumor suppressor and it's amplified in the tissue. I would say it's probably leading towards the likely benign. Like if it's an oncogene deleted in the tumor tissue, it's more toward that like direction as well. But for specific classification, I think we still need to collect all the evidence. So there's no like solid answer for this question, but I think that's something we want to take into consideration all the time, just to make sure we understand the molecular mechanism for each specific sample or each specific patient. well. Yeah, thank you.
Is there definitive detection of MCC by the prenatal array?
That is a good question. I didn't talk about the prenatal array today more thoroughly because we are focusing on the OncoScan and its function in the tumorigenesis. I think it's possible, but because I don't cover that part and it's more like a constitutional sample related, I would leave it for like more personal communication after the presentation to not just make the focus for this talk.
The next one we have here is FFPE tissue archiving time a factor for OncoScan performance?
That's a good question. So actually, there's a literature talking about FFPE tissue, how long. I think they trace back to almost 30, 40 years, and it's still working. But there's just like a few studies actually tested this. So I think it's really not a issue for OncoScan because the OncoScan performance is confirmed to be really good for samples like 40, 50 years old. We don't know anything about 100, 200 years old though. So that's the part I cannot cover, but I don't think it's a big issue.
Got it. Well, it looks like those are all the questions we have. So we'd like to thank Xiaolan Fang and our sponsor, Thermo Fisher Scientific. Thank you so much for joining us.
Thank you.